Hi, my name is Elodie Mitchell, and I worked at the Marsico Lung Health Institute in the Akuta Lab at the University of North Carolina at Chapel Hill this summer. And the Akuta Lab focuses on the lungs, and my project this summer primarily focused on the characterization of human small airway specific basal cells. And so, a little introduction into my project. So, the small airways, specifically the distal airways, comprise approximately 90% of the human conducting airway epithelial surface, and they represent the primary site of pathogenesis in mucoobstructive lung diseases like cystic fibrosis and COPD. And the small airway is also poorly understood on a cellular level because it's extremely challenging to capture the cells present in the distal airways, as the airways are extremely small in both length, from about less than 2 millimeters, and diameter with about less than 1 millimeter. And the distal airways are also molecularly, spatially, and functionally distinct from the proximal airways. And for these reasons, it's so important that more research be done on the small airways in our lungs. And so, some background into my project. The Akuta Lab has done a lot of research specifically on the differences between the large and small airways. And recent studies from about 2 years ago have identified specialized epithelial cell population, naming them terminary respiratory secretory cells, respiratory airway secretory cells, and terminal airway secretory cells. And the Akuta Lab likes to inclusively call all of these distal airway secretory cells and or DASCs. And DASCs are notably diminished in these muco-obstructive lung diseases, which is why it's extremely important to study them. And the Akuta Lab has done some research on DASC, specifically with looking at a lineage relationship between a subset of a small airway basal cells and DASCs in vitro. And to trace this DASC lineage to a specific subset of basal cells, they dissected some small airway epithelial cells, and they labeled them using an eGFP reporter, which is a green fluorescent protein under the control of the human SCGB3A2 promoter, which SCGB3A2 is a promoter, or it's a marker for DASCs. And they then differentiated these in culture and used something called FACS sorting, which basically just sorts the cells using fluorescence. And they did this sorting them through SCGB3A2, which I just talked about earlier, and then also NGFR, which is a basal cell surface marker. And some of the results of this study showed that this graph in the upper left corner, as you can see, which shows NGFR positive, EGFP negative cells, which are basal cells, most likely basal cells since they're just positive for NGFR. And then they also showed the blue marking on the graph is negative for both, which are most likely other secretory cells or ciliated cells. And then the red part of the graph shows NGFR positive and EGFP positive cells, which are most likely these DASC progenitor basal cells. And then they also saw NGFR negative and EGFP positive cells, which are most likely just these DASC population. And then you can also see they stained these cells using immunofluorescent staining, and you can see that the NGFR EGFP positive cells and then the NGFR negative EGFP positive cells were the only cells that show this like green fluorescent protein marking for DASCs. And then getting more into my project. So as you can see, the Akuta Lab was able to identify these basal subsets in vitro, but their in vivo identity and molecular markers that characterize these DASC progenitor basal cells still remained unclear. And so we hypothesized that NKX 2.1, which is a key transcription factor involved in specifying the proximal distal epithelial patterning, might regulate the differentiation process of specific basal cells into DASCs in the small airway. And so And if we were able to do this, the findings could advance our understanding of molecular mechanisms underlying these region-specific muco-obstructive lung diseases. And so to do this, I stained 11 slides using immunofluorescent staining for keratin 5, which is another basal cell marker, kind of like NGFR, and then also stained it for NKX 2.1, that transcription factor, and then for SFTPB, which is another marker for DASCs. And then I scanned these, and then I quantified approximately 65 airways from 5 different donors, counting up to about 200 cells on that epithelial surface of the airway. And then I measured the airway diameter using a device called QPath, and I measured them along the basement membrane of these cells. And then I also analyzed, and then I was able to analyze the data, as we'll see in a second. So the first thing I want to look at is this quantification process I went through. So you can see the proximal airway is on the left, and this shows staining of the proximal airway. And as you can see, the zoomed out version, you can really only see the keratin 5, which is that basal cell marker staining. But then if you zoom in on a specific part of the airway, you can see further that only keratin 5 was stained. Even if we isolate just the NKX 2.1 or SFTPB staining, you can only see, you see nothing on those. And then looking at on the other end of the spectrum at the respiratory bronchiole, you can see beautiful staining of this airway. And so I've isolated each one in the lower right-hand corner of keratin 5, NKX 2.1, which is that transcription factor for the small airway, and then SFTPB, which is that DASC marker. And then this last smaller picture just kind of shows you three different cell types that I quantified and looked for. So one of them being NKX 2.1 SFTPB positive cells, which is most likely those DASC cells under that NKX 2.1 transcription factor. And then you can also see keratin 5 NKX 2.1 positive cells, so negative for SFTPB, which is possibly showing those subset of basal cells in the distal airway. And then also the most interesting ones that I was able to quantify were the triple positive cells. So positive for keratin 5, NKX 2.1, and SFTPB, which is possibly showing these subset of basal cells that differentiate into DASCs. And so some data that I was able to collect from this, these are graphs that show and possibly show how NKX 2.1 correlates with the distribution of SFTPB positive cells. And so this first graph in the upper left-hand corner, you can see is only keratin 5 positive, so negative for NKX 2.1 and SFTPB. So these are most likely just our basal cells. And you can see, although there are outliers, there's a general trend decreasing as you go to the more distal airway, showing that these only positive for keratin 5 basal cells are diminished in the distal airway. And then the second graph shows NKX 2.1 and keratin 5 positive basal cells, but negative for SFTPB. So these are interesting because they're these basal cells that are just positive for that transcription factor NKX 2.1. And while, again, there are some outliers in the proximal airway, these are also mostly enriched in the distal airway. And then another interesting graph I like to point out is these cells that are actually negative for NKX 2.1, but positive for SFTPB and keratin 5. And those are only in the distal airway. And then the last one I like to highlight is the triple positive cells, which as I identified in the last slide, and these are also only enriched in the distal airway. Then another one I like to highlight is these pie charts, which are the average proportions of each cell type across the 5 donors that I quantified. And two important things I like to point out are the triple positive cells that I talked about earlier, which, as you can see these are the purple part of the pie chart. And while they are few as a whole, they're only start to pop up in the 1 to 2 millimeter airways, and then gradually do increase as you get to the more distal airway, the respiratory bronchiole. And then another one is this pink sliver, which is NKX 2.1 SFTPB. So these DASC cells that are highlighted under the NKX 2.1 transcription factor. And these are also only pop up around 1 to 2 millimeters and then increase as you get to the more respiratory bronchiole. And so what conclusions can we draw from this? So something important to note is that these keratin 5 positive basal cells co-expressing NKX 2.1 and or SFTBB are really only enriched in the distal airways. And these markers could be crucial for finding the functional roles of basal cells during both the developmental process and in these injury-induced and repair and regeneration in the small airway epithelium. And since this was just a summer project, future research should definitely be done to focus on identifying specific molecular markers that define the distal airway DASP progenitor basal cell population. And then I want to thank everyone at the Akuta Lab, specifically my PI, Dr. Kenichi Okuda, and then my bench mentor, Hiroaki Murano. Those are my resources. Thank you.
Done. Yay. Yes, look at all the motions. All right. Any questions or comments? We have time for two, if anyone has any. Okay. I guess the same question I had before, and I like to always ask is, what was your favorite part, favorite thing you learned, or favorite like experiment you got to do?
Yes, so my favorite thing was actually I came into this not knowing much about the lungs in general or um, basic like lab techniques. I hadn't done much of that at all, and so this internship was amazing for getting to learn all of that, but also getting to apply it, and then being able to do this project and get results that hopefully will help the Akuta Lab in their future research was extremely interesting.